So hi, my name is Denis Jabaudon. I'm a professor at the Department of Basic Neuroscience at the University of Geneva. I'm also an attending physician at the Department of Neurology at the HUG. So I'm trained as a medical doctor. I did my medical studies in Lausanne and then did a PhD program at the University of Zurich, where I got interested into synaptic physiology and circuit connectivity in the hippocampus. For that, I did my internal medicine residency and neurology residency, and then went for a postdoc at Harvard in Jeff Macklis's lab, where I got interested into neuronal development. Came back to Switzerland in 2008, where I started my own lab trying to bridge together my interest in circuit and synaptic physiology and development. I have a 20% clinical commitment, and it's true that in my case, my research activity has no direct relationship with my clinical activity. I think it's a strength in the sense that it gives me two perspectives to ask questions from, and also from the point of view of the clinics to get a realistic point of view as to how drugs or therapeutic approach could be translationable, and from the basic research point of view to get a realistic understanding of what we actually can do. I always was interested in basic research, and I always wanted to have the bigger picture. As I was interacting with patients at the hospital, I really felt the need to go a little bit deeper into the mechanisms of what was going on, and that really drove my research interest. You know, compared to other fields of medicine, the exciting part is that you can study the same organ from a very detailed molecular point of view to a more cellular point of view, but then also to a network and then cognitive point of view. And I think this is what's exciting about the field that we're in, right? You can interact with very different kinds of people with very different backgrounds, and that brings a lot. Yeah, so we're interested in the development of the cerebral cortex, and more specifically, we're trying to understand and identify the genetic programs that generate neuronal diversity. The cerebral cortex in particular is a cellularly highly heterogeneous structure. There's neurons that project from one place of the cortex to the other, that projects to the spinal cord or other brain regions. And these are not only very specifically wired, but can be also defined by their gene expression. And so we're trying to understand how from a seemingly relatively homogeneous pool of progenitors this diversity can emerge. Not only in the direction of how do genes control the generation of different neuronal types that control the generation of different types of circuits, but also in the other way around. How does circuit identity, how does the fact that a given neuron belongs to a circuit influence its gene expression? It's a two-directional interactions. Genes influence the building of circuits, but the wiring of circuits influences the expression of genes too. Well, I think it relates to the interactions between genetic and environmental factors. So in that sense, I would say the most direct relevant are things that relate to susceptibility genes and interactions with the environment, so probably psychiatric disorders or neurodevelopmental disorders. But of course, there's more and more evidence, or at least, you know, the field is shifting towards a point of view where the cell death that occurs at relatively late ages in neurodegenerative diseases might be the reflections of susceptibility factors that are present very, very early on, maybe during even embryonic or early postnatal development. I guess I'm learning a lot from these patient cohorts types of approaches that I'm not particularly familiar with. So in the same way that I was saying a couple of minutes ago that it's important that the clinicians learn how things are going on in basic research, I think it's important for those of us who are not directly exposed to that to understand the limitations of the cohort approaches, the diversity of the patients, the type of information that can be gathered even from single case studies as proof of principle examples of what can happen in a disease. So yeah, it's quite rich, and again, it's developing a common culture. The development of such a common culture in the neural world in particular, but I would say in medicine in general, in medical studies, is absolutely critical and undervalued in my view. I mean, a good way to promote scientific advances is to promote human interactions, very simple human interactions, so that people feel comfortable with one another and confident about what the other is saying. And I think Synapsy does that in a remarkable way.